Okay, great. Thank you for that introduction. My name is Ariana, and today I'm going to be talking about the security officers in your heart that could be a key to curing heart disease. So before I get into talking about heart disease, which I know is not the happiest topic out there, I want to start out with a piece of good news. Life expectancy is growing dramatically. So we can see in this graph here that in the let me get my laser pointer. In the mid to late 1800s, people in the US were only living to about 40 years old, but now we're living to almost 80. This is very exciting. It's greatly due to the fact that a lot of scientists and doctors have done work to better understand how to both prevent and treat different illnesses so that people can live longer, healthier, and hopefully happier lives. And we're likely all very grateful for this. I am definitely very grateful that I'm not already past middle age. But there will always be new issues to tackle as we start to prolong this lifespan, and one of those main issues right now is heart disease. So heart disease is currently the leading cause of death in the US, outranking things such as cancer, accidental deaths, deaths due to stroke, and more, and resulting in about 700,000 deaths per year in the US alone. Now, unfortunately, this issue is likely going to get worse before it gets better. And this is because in the US, we have an aging population. We're starting to see the number of older people outweigh the younger, and we also have an aging baby boomer population who are starting to have some more medical needs. So our health system is expected to be increasingly burdened by the medical needs of this aging population, and this means that there will likely be more and more cases of heart disease that will need treatment. So this is where I and many, many other scientists around the world come in. We are trying to learn more about what risk factors are causing this heart disease, how these risk factors cause heart disease. So not just knowing that like smoking can lead to heart disease, but what are the actual mechanisms in the middle there, because that will help us to be able to better create new treatments for heart disease. So we need a way to be able to answer these questions. We need to be able to test out our hypotheses to improve our understanding of heart disease. But we obviously can't just test this all on humans. I don't think anyone here wants to volunteer for me to take you back to the lab and try and give you heart disease and run some tests. So unless I get any takers on that tonight, and just come see me at the end, testing all of our heart disease-related hypotheses on humans is probably out of the question. So we need to figure out which elements of the heart are really critical to studying heart disease. So maybe we can take just those parts and try and study the heart outside of the body. So what are these critical elements of the heart that would be really important to include? Well, the first one has to do with something that you probably think of pretty readily when you think of the heart. It's beating. So heart muscle cells are the cells hard at work all day long, keeping your heart beating. So these are very important for heart's function, and we can imagine that if the heart's not functioning properly, something has probably gone wrong with these cells. The next thing that's important that you might not think of as readily is the blood vessels in your heart. So when you think of blood vessels, you probably think of the blood vessels that run all throughout your body, bringing blood to your different limbs and organs, but your heart also has a ton of blood vessels within itself. And these blood vessels are really important for supplying the heart muscle cells with all of the oxygen and the nutrients that they need. And to give you an even better understanding of really how critically important these blood vessels are for the heart's function, here's an actual image of a piece of heart tissue. And here we can see all of the heart muscle cells outlined in red. So all of these bigger shapes here are our heart muscle cells. And then we can see all of the blood vessels outlined in green. So all these little circles here in this greenish yellow color are our blood vessels. And we can see that there are a ton of blood vessels in comparison to the number of heart muscle cells that are present. In fact, it's said that each heart muscle cell is in contact with at least one blood vessel at any given time. So these two elements have a really important relationship in being able to help each other and help in heart function. We have, to kind of recap, we have our heart muscle cells that are keeping your heart beating, and we have our blood vessels that are providing those cells with all of the oxygen and nutrients that they need. And now I want to dig a little bit deeper into understanding those blood vessels so that we can see that the role they may have in curing, but also possibly causing heart disease. So here is an example of one of those many blood vessels that runs throughout your heart. And in this cross-sectional view here, we can see that there are these endothelial cells that line the inside of our blood vessels. And to give you a better understanding of what these endothelial cells do in the heart, I want you to think of your heart like an office building. So this here is your heart. And within that office building, you have your workers. In this case, it's our heart muscle cells. So they're constantly hard at work, keeping your heart beating. And you want to make sure that they are able to do their job. You don't want them to be in any way disturbed because your heart beating is a pretty important thing for you surviving. And so because of this, you put some security officers or endothelial cells outside the building to protect the building. So those endothelial cells or security officers are going to control what is and is not allowed to enter the building and interact with those workers. So for example, if someone shows up to the office building with a shipment of water, those security officers are going to go investigate. They're going to decide that that water looks like it's really important for the function of those heart muscle cells, and so they're going to allow it to go into the building and go visit with the workers. However, if instead someone shows up to the office building with vials of toxins, the security officers are going to go investigate and they're going to say, no, that looks like it would be really harmful for the workers. You are not allowed to enter. So this is pretty much how it works within your heart. I made this little diagram here where we can see the heart tissue here in this red color. All throughout that heart tissue, we have our heart muscle cells. And then going through this heart tissue, we have many of these blood vessels, as I showed here in this lighter pink color. Through that blood vessel, you have your blood flow that's carrying all different elements through your bloodstream. And then very importantly, lining this blood vessel is these endothelial cells, and these create this really important barrier between your blood vessel and your heart tissue so that they can control what can go from your blood vessel to your heart tissue and what cannot. So for example, if they see something useful traveling down the bloodstream and they think that would be useful for your heart muscle cells to function, they will allow it to pass through that endothelial barrier and get access to the heart tissue. However, instead, if they identify something as non-useful or even harmful, they will not allow it entry. So this is how your heart functions when it's healthy. There's this really important relationship between your endothelial cells and your heart muscle cells. But what happens if something goes wrong? Let's say one of your security officers gets sick, and soon he passes it on to a lot of the other security officers, and before you know it, almost all of your security officers have called in sick. There's no one left to protect that office building. Well, this can actually happen within your heart. So your security officers can become sick, or your endothelial cells can become sick for a number of different reasons and basically just not report to work to do their job. And this can be really dangerous because it means that, maybe those useful things are still able to pass through and get access to the heart muscle cells, but maybe some of those non-useful or even harmful things are able to find this gap in the endothelial barrier and get access to those to that heart tissue and to those heart muscle cells. And to make matters even worse, some of the endothelial cells, maybe they haven't died off, maybe they're still trying to do their job, but they're very sick. And this means that, normally they're sending all kinds of healthy signals to the heart muscle cells, telling them everything's going good, we're doing great, we're protecting you. Instead, they're sick, they're kind of sending off these very confusing signals to tell the heart muscle cells that everything is not okay. So they're telling the heart muscle cells things such as get inflamed and die, and form scar tissue, and get really big, and do crazy things. So why this is also important and why I took a little bit of a deep dive into better understanding the role of blood vessels in your heart is that scientists have actually found that this dysfunction of the endothelial cells can lead to heart disease. So this provides scientists with a whole new set of questions, such as what causes this dysfunction of the endothelial cells in the first place? What exactly is happening to these heart muscle cells when endothelial cell dysfunction occurs? And then lastly, and possibly most importantly, what treatments can we use to cure this dysfunction, and can they also cure the heart disease that may have occurred as a result? So, in order to answer these questions, let's go back to the important elements of the heart that we want to include when studying the heart outside of the body. We now know that the heart muscle cells are the main functional cells in the heart, keeping it beating. We also now know that the blood vessels in your heart and the endothelial cells that are lining them are playing a really important role in both getting oxygen and nutrients to your heart muscle cells, but also in protecting them. And we want to learn why the endothelial cells that line these blood vessels sometimes don't do their job correctly, leaving the heart at risk for heart disease. So, how can these two important elements be combined outside of the body to learn more about the role of endothelial cell dysfunction in heart disease and how we can treat it? Well, in the past, scientists have studied cells and diseases in a number of different ways. One of the most common things they have done is use something called 2D culture. And this is when you basically take your cells of interest and you put them onto a petri dish. So for us, we would take our heart muscle cells and our endothelial cells and put them together on this petri dish. And then we can kind of study, how they're interacting with each other while they're on that dish. We could also try out some different experiments. For example, we could put some solution on there that should make the endothelial cells sick and see what effect that has on the heart muscle cells. We could apply some drug that should be more curative and see if it has that effect on the cells. However, while this is likely the simplest way to study how these cells interact, it doesn't necessarily reflect how they actually interact in the body. And this is because our bodies are not 2D. We are not all flat Stanleys. So our cells are used to being able to stretch out and move around in 3 dimensions. And without this ability, they might be a little uncomfortable and a little bit unhappy. So you can think of this like having to sleep on the floor instead of in your bed. So you're not going to be comfortable, you won't get a good night's sleep, and because of this, you might not act quite like yourself. And so this has caused other scientists to kind of take a step further and do what is called 3D culture. Now, there are a number of different ways this can be done, but I have one example of this here. And here the cells are inside of a 3D environment of collagen. And collagen is a structural protein that makes up the framework or scaffolding for many of your different organs, including the heart. So these cells are used to being in this collagen environment. So they're now able to stretch around and move around in multiple different directions. So this is much more like sleeping in your actual bed. While this might make your experiments a little bit more complicated, your cells will likely be much happier and act more like they would within your actual body. So I showed you a couple methods scientists have used to study cells and diseases in the past, but unfortunately these methods have one main issue when it comes to studying heart disease and the effect of endothelial cell dysfunction on heart disease. And that issue is that they are lacking this one important element. And so in order to figure out what that is, let's look back at this diagram I showed you earlier. And we can see, all these different elements that we want to make sure to include when studying this. And the 3D culture methods have kind of represented this heart tissue by using that collagen I talked about. So they kind of checked off the heart tissue box. They also, both of those past methods allowed you to include multiple different cells. So you could include your heart muscle cells, you could include your endothelial cells. But the one thing that's really missing from what has been done in the past is these blood vessels. So both those 2D and 3D culture methods don't actually incorporate any blood vessel. And this is important for a couple different reasons. One, because if you don't have your blood vessel, you don't have blood flow, and your body is normally functioning with blood flow, so this is going to be pretty representative of what's really going on in your heart. But also, if you don't have a blood vessel, then you don't actually even have this endothelial cell barrier. And when we're studying endothelial dysfunction, we're really interested in what's happening to that barrier, when things are able to kind of sneak through it. And so if we just have random endothelial cells spread throughout a plate, they're not able to form that wall around a vessel. So this is really important to include. So this is where my work comes in. I am creating an improved way to be able to study endothelial dysfunction in heart disease. So in order to do this, I started out with a list of criteria I wanted to make sure to meet. And the first criteria I set was that I wanted to make sure I was making something that would include a blood vessel element, as I just kind of described there. I also wanted to make sure that I was able to apply blood flow through that blood vessel. I also wanted to make sure that even though I'm adding in this blood vessel, I'm still including those important cell types, the heart muscle cells and the endothelial cells that are really critical for me being able to study this problem. And then lastly, even though I'm making things a little bit more complex by adding in something new, I still want to make this as simplified and easy to produce as possible. Because I'm likely going to have a ton of hypotheses, many of which are wrong before I get to any solutions here. So I want something that I can make pretty easily so I don't go too crazy in the lab. And so what was the solution that I came up with here? My solution is what I call rigatoni tiny hearts. And if you need a refresher of what a piece of rigatoni pasta looks like, this is a piece of rigatoni pasta. It's little tube-shaped pieces of pasta, and I call what I make rigatoni tiny hearts because they're tiny little heart pieces that should kind of resemble the heart's function, but they're also shaped like a piece of rigatoni. And before I show you the actual images of what my rigatoni tiny hearts look like, I want to tell you a little bit about how I make these. So just like making rigatoni, or any other pasta, the first step is making my dough. But instead of using actual pasta dough, I use something I mentioned earlier, collagen. So as a refresher, this collagen is a structural protein that's kind of making up the framework or scaffolding of your heart. So these cells are used to being in that environment. So this is what I use for my dough. And I mix in my heart muscle cells all throughout. Once I have my heart muscle cells mixed all throughout my collagen dough, I use a mold to form this into this rigatoni shape. And the reason I form it into this rigatoni shape is because it looks like something I showed you earlier, a blood vessel. So the next step in making my rigatoni tiny hearts is to line the inside of my piece of rigatoni with endothelial cells. So what I basically have now is this little piece of heart tissue with a blood vessel running through it. And I can even apply some media or blood flow through that hollow center of my rigatoni noodle. So what does this actually look like? This here is a video of my rigatoni tiny hearts. Oh, if it will play, maybe not. Okay, well, I'll describe it to you here. It just zooms in a little bit. So here you can just see in relation to my hand, and each of these little pieces here is a rigatoni, or is one of my little tiny hearts. So here's a tiny heart, there's a tiny heart. These little black pieces on either side is just where it's sutured to these needles that hold it up. And one thing you can't tell from this is that these are actually hollow needles. So I can actually like hook this up to a syringe and flow some type of solution through. It will go through my little tiny heart that's here, and it will flow out the other end. So now I'll show you some zoomed in images so you can get an even better understanding. So first, this is an image just under like bright field microscope, which basically just means it's shining a bright light onto my tiny heart. And here you can really see how it has this hollow interior. So this is where my solution can flow through. And then on either side of the hollow interior, I have these walls. And then I kind of wanted to show a better image that really showed what this looks like in 3D, because this is kind of hard to visualize in just like a 2D way. So then I put cells within my tiny hearts, and then I put markers on them that basically make it so if I shine a laser on them, they will glow in the dark. And so I was then able to get this fancy image with a fancy microscope where you can really see how this kind of does look like a piece of rigatoni pasta once you zoom in on it. And then I also wanted to show, and maybe this video also won't work, we'll see. I think you can see a little bit. So here, let me go back. I have these beads that are flowing from one end to the other end. It's maybe a little hard to see with these lights shining on it, but and this means that I can flow something through from one end of my tiny heart and it will flow out to the other side. So, does this meet all of the criteria that I had initially set? Yes, this does include a blood vessel element. It also allows us to apply blood flow through the center, as you could maybe see from that bead video. It also allows us to include heart muscle cells and endothelial cells, as I showed in this schematic up here. And then lastly, you're gonna kind of have to trust me on this last point, but I will say compared to a lot of the other things I have made in the lab, this is pretty simple and easy to produce. I'm able to kind of mass produce a ton of them, which is very, very nice. So I showed you these rigatoni tiny hearts I made. What's next? Right now, I am continuing to perfect my recipe, trying to work on getting my Michelin star, if you will. And I have something that works, but it could still be better. I'm still doing a lot of troubleshooting. I'm also still trying to work to make it even more similar to the actual human biology than what I have right now. Because in the end, I hope that this new method for studying the interactions between heart muscle cells and blood vessels will help us to learn more about what is causing our heart security officers to get sick, what effect this has on the workers or the heart muscle cells, and what treatments might work to be able to solve this problem. Thank you.